Welcome to the Bioinformatics Chat. Today I'm speaking with Andrey Ziyatdinov. Andrey is a postdoc at Harvard School of Public Health, and he recently published an R package and a paper called lme4qtl, linear mixed models with flexible covariance structure for genetic studies of related individuals. Andrey, welcome to the podcast.
Yeah, thank you, Roman. Thank you for inviting me. Actually, I looked through your previous podcast and I was really interested in a diverse topic you cover in your bioinformatics podcast. I learned many things and I'm happy to be here.
Yeah, and we didn't have anything, I think, about, definitely not about mixed linear models and very little about QTLs. So this is going to be a good addition. Let's talk about yourself. What is your background? Where did you study? What did you study?
Yeah, sure. So my basic background is in applied physics and mathematics. I got my bachelor degree from Moscow in Institute of Physics and Technology. My PhD was in biomedical engineering, not too much about genetics, but I got this kind of statistical and technical background. And 5 or 7 years ago, I started to work in this field, let's say bioinformatics, genetics, and statistical genetics. And after a stay in San Pau Hospital in Barcelona, where it was pretty clinical studies, so I moved to Harvard, Harvard School of Public Health. And here I'm in a program of genetic epidemiology and statistical genetics. And here we have method development and also analysis of large-scale data available here within Harvard or globally.
How did you come into bioinformatics? What brought you from applied math and physics to genetics, say?
Well, my PhD advisor has this kind of several lines of research, and one it was more computer science engineering when I was actually trained as a PhD student. And the second line it was bioinformatics. And in Barcelona there are many hospitals and research institutes. And I moved smoothly to this area, and it was boom of availability of genomic data and any kind of omics data. And I was really amazed. I remember I took some courses at Coursera, this online platform, and I started feeling that I know more about biology and maybe I can apply my, let's say, mathematical statistical skills to biomedical genetics problem. And it was pretty successful. So I think I was thinking maybe it's good to keep going. So that's how I appeared here in Harvard.
That's awesome. And so your lme4qtl package, is that your first sort of major contribution to this field, or what did you work on before?
Actually, I would say this kind of R packages, this was side projects, because when I started this technical project, I was actually involved in analysis of clinical data in that hospital in Barcelona. And sure, I collaborated with all the people and I provided them statistical, analytical expertise. So we worked together and most articles published was application of standard method or method we developed within the lab. And one of that packages was this lme4qtl. And yeah. And actually, I finished that project and published the article. Being here in Harvard, it took 5 years to finish it. So I'm not sure it's a long project, but finally it's already there, and all the people can use it. People from the field are pretty interested in it.
So your package, lme4qtl, it's a package to analyze QTLs, right? So let's maybe start with this. What is a QTL?
I guess many of your listeners heard about GWAS, which is genome-wide association studies, which was last 10 years really booming, and it's mostly applicable and kind of reliable to get new insights on genetic architecture of diseases and complex traits. So the idea is if you have a clinical studies and you have phenotypes, let's say can be a quantitative body mass index or height, or can be a disease status, having disease or not, binary data. On the other side, you also can genotype your individuals, get a single nucleotide polymorphism data, So it's like millions of SNPs. So like next, the question is just to do association study, pick one genetic variant at a time, just test. Basically, it's kind of correlation between your phenotype and genotype, and do it so many times you have your genotypes. And then you can just see what the contribution of top genotypes or like generally like what look like the shape of variation of phenotype affected by genotypes. And when we say QTL, is abbreviation stands for quantitative trait locus. So it means a particular genetic marker in the genome that has an effect on QTL. Maybe 20 years ago, genetic association studies were mostly done in families because they have this kind of different study design, and they still didn't have access to this large scale and high resolution genotyping arrays. So they just took what's called microsatellites, which was like hundreds of genetic markers, and they did linkage studies. So I guess this QTL notation came from that times. And like last 10 years, now people do genetic association studies, GWAS, but still QTL is the same idea that you looking for particular sites of genomes that have something to do with your phenotype.
Let's start with just a simple linear model. How could we apply a simple linear model to a GWAS study?
Yeah, in a nutshell, you have a correlation coefficient like between genotype and phenotype, and you can perform a statistical test and get a p-value. If you do it for like millions of SNPs, you have like many p-values, and so you're faced with a problem of multiple testing. And so you apply typically Bonferroni threshold, and if you have enough sample size, so your study is well powered, and you can actually observe some peaks, which, and if you make a plot where on the x-axis you put like genomic position, on the y-axis you put like log 10 p-values, so you have kind of Manhattan plot. That's what you can finally get. But linear models, it's a good thing because you can add covariates. Let's say complex diseases are affected by environmental clinical factors and as well as genotypes, so you likely want to take into account the effect of age, gender, like many other like related covariates. So linear models are good for that. And you can also, like if you have binary outcome or even like some count data, you can use generalized linear models, and you still can perform the job of the association study. So this is like linear models, but what people observe that when they like early started with GWAS, that you can have inflated statistics, meaning that you can be too optimistic, and they were thinking how to deal with that, and you can have some structure in the data. If you have homogeneous data from one particular location and there is no like family relatedness, they're all independent, linear models will likely be the good choice. If you have some, let's say, even within Europe, you have like some people from the south, some, let's say, Spain, other group of people from France, so you have some population structure, and it's part of the field of population genetics, and people were thinking how to address this problem, because if you perform a test without controlling for this population structure, you will likely find SNP, let's say, that distinguish like eye color between Spanish and French people, and not these kind of genetic variants associated with the disease. So there are many methods to address this, to model this population structure, and one of them is mixed models.
Right, so in a simple linear model, you would include as your covariates, as your variables, you would include both things like age and sex and height or whatever, and you would also include the genotypes, right? So how does that part work? How do you incorporate your genotyping data into a linear model?
Yeah, so as I said, you pick one genotype variable at a time. So, and let's say you have like B allelic variant, you can convert these labels to numbers. Let's say if you follow this additive model, you will end up with 0, 1, and 2, meaning like the number of minimal allele in a genotype of a particular person. So in this way, for every SNP, you have a vector of the length of the number of your individuals in the study. And like each entry in this vector is 0, 1, or 2. this is your quantitative predictor to test, and you just include this vector of genetic variant into a model, like a new predictor, feed the model, see the like effect sizes and the error, so you can compute a test statistic, which is like can be t-test, or anyway, it's pretty similar, and so you derive a p-value.
So you would fit this linear model for every single SNP that you genotype, right?
Yeah.
And the same issue applies with multiple testing, so you still have to apply the correction to the resulting p-values.
Yeah. So people several years ago already established a number of, let's say, independent genetic markers, because genetic markers close in location, physical locations, they kind of have correlation structure, so they're not independent. And like it's estimated that you have a million of independent markers. And usually it's not like you can genotype like 10 millions of SNPs, but finally you end up correcting for 1 million, because that's like give you a Bonferroni threshold, which is 5, 10 minus 8, I guess, is a kind of standard threshold and when you consider for multiple testing.
And I guess if you have not a biallelic SNP, but when you have more than 2 alleles, you could introduce this as separate covariates. So you would have a covariate for allele 1 and for allele 2 as compared to, let's say, allele 3 as a baseline.
Yeah, actually, I've never worked with 3 allelic or something, because like for biallelic and for human traits, it's well established that additive model is a good, let's say, transformation from your genetic data to quantitative variable. And like even for biallelic, you can have, in addition to additive coding, you can have some dominance. So everything which is violates this like additive model, like linear one, and you can include this. But what happens in human genetics that people know that contribution of this dominance effect of like SNPs, a single SNP or like more or less genome-wide, is really a way less than contribution of additive effects. So people just do additive coding and in most cases and just perform this GWAS in these settings.
Right. So an additive effect is when the jump from 0 to 1 of the minor allele brings the same effect as the jump from 1 to 2.
Yeah, you're right. So this is like kind of model. If this is not true for some, imagine extreme cases. So then you can have an option to include dominance effect, and you likely find a particular genetic variant that manifests this kind of nonlinear like dominant effect. Yeah, but in most of the cases, you simplify, and in real life, additive model works pretty well.
You mentioned that if we don't account for population structure, so people in Spain and people in France and people in Spain are similar to one another and people in France are similar to one another. And so if we don't account for that in our simple linear models, then we'll run into problems. So what kind of problems? You mentioned we might find a spurious locus that would account for the eye color. So why exactly and how exactly would that happen, and why wouldn't we find the true loci that account for the eye color?
Yeah, so I would say going to like basic linear model, that imagine that you have, forget about genetics, and you have some like outcome you want to model. And for sure, if there is some important covariate you don't include in your modeling, so you just having like you can get some biased estimates or biased effects of data you have available and measure it in your data set. So this is something particularly applying to kind of linear models in genetics. If you know there is population structure and you don't account for it, so you will likely get something which is far from the true laws of this phenotype that shapes this phenotype. And I guess when I say this kind of eye color, I remember in the course I took in this Coursera, it was a example when like a person couldn't publish his article about GWAS results because like in a good journal, and they suggested just you should to do something with your population structure, and he didn't took this advice and published in a low impact journal, but still it kind of resulted were very suspicious.
So I'm curious, would it help if instead of running a linear model for each separate SNP, what if we included all the SNPs and then more important ones would get higher coefficients than less important ones?
You mean instead of including one SNP at a time, you put everything, all SNPs, right?
Yeah, so that those SNPs that are just correlated by accident, presumably they would have a weaker effect than those
Yeah.
that are truly causal.
Yeah, actually, I should explain that people really do something similar. So if you have like some individuals and it can be some structure on them or not, you don't know. And the way to model it is to construct so-called genetic relationship matrices, where you use your genotype data and compute based on this genotype data, which are usually centered and scaled. So you compute the relationship matrix. And this matrix appears to model very well how close a given pair of individuals based on this genetic population structure. And exactly like mixed model, the framework allows you to include a random effect with this variance covariance matrix, which is like GRM, and that helps you to model the global effect of all SNP shaping like populations and like include it, let's say, not as a covariate, but as a random effect, but still you control for it. And then you are safe to like adding SNP at a time in the presence of this genetic relationship matrix, and you just testing and you can like collect your p-values. And later on, people usually do this quantile plot because you expect a uniform distribution of your p-values because like it's a hypothesis-free study. This is the way people currently perform GWAS studies.
Yeah, I like how you segued into mixed linear models. Okay, so tell us about them. The things probably that we want to start with are, what are these random effects and mixed effects as opposed to fixed effects? What do these all words mean?
Yeah, so if we do a step back and think about linear models, we have like outcome predictors, and all these beta coefficients are like kind of fixed effects. So like you don't assume any distribution on that. And at the same time, with the simple linear models, you have a residual term, right? Which actually it's a random effect. So you just assume that it's a effect that distributed, which is called IID, identically along individuals. And like it's something that typically refers to a noise term, but actually it's like random effect of which has a normal distribution. And once imagine you have some, let's say you collect data from different medical centers, right? And you likely know that there is some effect of this medical center, but it's not kind of causal. It's just some people are different based on whether they belong to one medical center or another. And you can include this as a random effect and just based on this grouping matrix. And that helps to take into account this kind of randomness due to, let's say, grouping center and run linear mixed model and do kind of this accurate modeling. In terms of genetic studies, if you model, let's say, families or population-based studies, there is some genetic relationship between given pairs of individuals. And again, you can collect the genotype data and compute this kind of relationship matrix. And yeah, these random effects actually can be like kind of genetic, what I said, or can be, let's say, grouping effect, or can be, imagine you have heteroscedasticity effect, meaning that there is some difference in accuracy you collect data. Imagine like you have like two genotyping arrays and each one can have good quality and low quality, and maybe errors are not equally distributed. So you should just precisely model this kind of two noise terms. And like, again, mixed models are a good instrument, like good framework to do that.
So you mentioned this example where you have two medical centers and you assume that there might be some difference due to the difference in medical centers. How is a mixed linear model different from including this medical center as just another covariate in your simple fixed effects linear model?
Yeah, it's a very good question because I guess in most of publication, people, as you said, included a covariate. So a short answer that for the purpose of testing association for a given SNP, let's say, so it's equivalent because somehow the variance due to let's say medical centers, you can model equivalently by one way or another or the other. But I guess if you're interested in, let's say, be more precise in quantifying the effect of belonging to one center or another, so these kind of random effects, they have kind of shrinkage. So let's say you have like 5 centers, right? And if you include the covariate, you will have 4 coefficients. You should encode like dummy variables, and you have like a baseline and like base, and then you have like what's like additive effect of belonging to one group comparison to the baseline, and then to the third group, and to the so on. So we have like 4 coefficients. And if you model as a random effect, you still will have 5, let's say, effect sizes. But in this case, you'll explicitly assume that these effect sizes have a normal distribution, and it will be kind of more, let's say, plausible assumption that if you model as a fixed effect, you might have not enough data to model this strong assumption that they're all independent. And in most cases, you will have larger magnitude of these effect sizes if you model as fixed effects. But as a random effect, you will have kind of shrinkage. And this is kind of quantitative differences.
Yeah, this is a very good point. So how does this then relate to Bayesian linear models? Because this sounds like just a good Bayesian practice where you put priors on your parameters that you try to estimate. So is this just a special case of a Bayesian linear model?
Well, yeah, we know that like there is a link between Bayesian and like frequency statistics, right? Yeah, in this particular case, there is a link because, like, if I guess a prior distribution for fixed effect is kind of uniform, right?
Or you could even put, priors on your fixed effects. It's just a good practice because there too you have some prior assumptions.
Yes, I'm not very proficient in Bayesian modeling, but as far as I remember, if you try to match like your frequency approach, like running linear models or any more complex model, and like Bayesian approach, the way to get the same results for linear model and like fixed effects is to put, I guess, uniform prior on fixed effects. So and you will end up with the same results. And yeah, in case of random effects in framework of linear mixed model, so you explicitly say that distribution of your effects, let's say due to the center or like any other grouping factor, so it's kind of normally distributed. And knowing this, so you can put it all together, actually, like all random effects are normally distributed, and that gives you like your phenotype or your outcome you model is actually like multivariate normal. So we can apply maximum likelihood, construct this maximum likelihood function and just optimize and like get estimates of this kind of contribution of random effects.
Yeah, so to summarize, random effects differ from fixed effects in that first, they kind of have priors on them in this frequency setting. And the second, I guess, the difference is that you don't really get from the model the estimate of the random effects. So random effects are supposed to be the effects you're not directly interested in.
Well, one thing, you get estimates of your random effect, which are conditional on your kind of structure due to fixed effect, because it's kind of conditional distribution. So it's one point, and that, yeah, it's something like not true estimates of random effects. In this sense, you're right. But still, using this kind of conditional estimates, people like do use this estimation of effect sizes to do kind of prediction. I guess in like agricultural science, yeah, they have this inbred species, and they still like feed models, estimate this random effect, genetic random effect, and build prediction models based on this kind of even kernels or this kind of more like genetic relationship matrix. So I would say more they develop these prediction techniques based on estimation of random effects. Maybe, I don't know, many years ago, then like in human genetics they start to apply this method.
So in your example that you gave, if you have 5 medical centers and you want to model them using random effects, in that case you would have 4 random effects, right? In addition to the fixed effects?
Well, you will have 5 actually. Yeah, because like if you model as a fixed effect, you have intercept which is kind of included is one of the effect of the center.
Right. And then if you want to apply this model to genotypes, then you would have to include 1 million of random effects, which is clearly wouldn't work. So how do you apply these mixed models to GWAS?
Yeah, when you estimate this random effect, you model in distribution, right? So you model in distribution of either given like 5 or million of distinct, let's say, SNPs or any kind of grouping. So and what you estimate, you like first you assume this is like normal distribution, right? So like normal distribution with mean 0 and some variance. So what you get for every random effect, either it's million of SNPs or 5 medical centers, you get this sigma parameter, standard like variance. And once you get it, so you can just get a good modeling of your variance of your phenotype, which depends on like grouping factors or genetic effect. That's the way you get estimation of random effects.
Yeah, this is something I wanted to clarify, because in mixed models, there are two types of covariance matrices that can be considered. So you can either consider the covariance matrix of your random effects, because they are modeled as a multivariate normal distribution, or you could consider the covariance matrix of the samples themselves. And if you have the covariance matrix of your random effects, and then if you know which samples are affected by which effects, then you can come up with, from that presumably smaller covariance matrix to this bigger covariance matrix that covers all of your samples, right? Is that a right way to think about this?
I kind of guessing what you're asking, though, so let me try to give you. Imagine like that example, you have like a clinical study, you collected like a phenotype and genotype from these kind of 1,000 individuals, and you have like 5 medical centers, right? So when you include like 2 random effects, which one is due to medical centers and the second due to like genetic data. And so in the first term, you will get 5 by 5 covariance matrix, because you have like 5 groups of individuals, right? Due to 5 medical centers. But in the case of genetic random effect, so your covariance matrix will be like this number of individuals, like what I said, like 1,000 by 1,000, because you are interested in modeling like each pair of individuals. Because like let's say, like in some particular case, if you have like families, right? You have parents and their children. So for sure there is some like people know like how they the proportion of genetic variants they share. Answering your question, you have, depending on whether you're modeling grouping to the center or like genetic effect, which is like individual level similarity matrix. So you'll get different size of covariance matrices.
Yes, and not just different size, but two different types of covariance matrices, right? If it was a single way, then in the second case, you would get a million by million covariance matrix that would describe your individual SNPs, but that's not what you want.
Yeah, here what you're modeling, you're right. You like your size of covariance matrix, like number of individuals and not number of SNPs. You just use all your genotype data to estimate this per individual level relationship in case of like genetic similarities.
And so I'm guessing that this is related to what you did in your package, right? So you had to implement this second way of parameterizing a mixed model?
Yeah, since you notice that like this R package is an extension of another R package which is called lme4, which is relatively widely used in not only genetics field, but in many like psychology or like ecology and many other fields. So, and what I wanted to do with that package, just to make use of all infrastructure created for like last 15 years or something like that, and apply to like genetic studies. But it happened that it was not possible to add some like custom covariance matrix, because for when we do genetic studies, you have genotype data, you construct this genetic similarity matrix, and you just want to include them as in a mixed model. But what I encountered that lme4 didn't allow you to do that like that time, and I guess like with the last version that appeared like a week ago, they don't support this kind of functionality. And since in genetic studies, including a custom covariance matrix is really like a common thing to do, so I was thinking just to do some kind of coding and just see how it was implemented, maybe implement some additional matrix operations for doing that. And finally, it was useful for me and like for all other people that doing this kind of similar studies.
So when you fit a mixed linear model, you have to do several things. So for example, this lme4 package, it has to estimate the fixed effects, the random effects, and the variance or the covariance or the random effects, right? And when you supply your own covariance matrix, you're essentially reducing the amount of work that it has to do because you're saying, well, don't bother estimating the covariance matrix because I think I know what it is exactly.
Yeah, you're right. I guess you're getting the point that if you want the mixed model framework estimate every particular pairwise like entry in covariance matrix as well as like contribution to every let's say random effect with its covariance matrix, it's a very hard job. So what like a traditional way to do that is to say that I know the structure of my random effect. Let's say that can be like a genetic relationship matrix that you estimated using genetic data, or if you know that it's 5 centers, you say, oh, there is only 5 centers. So first step of this running linear mixed model is to estimate what is called variance components. And in particular, this kind of, you encode this covariance structure as a function of really few parameters, which is like basically this sigma squared, or it can be correlation coefficients. So, and usually like in the simplest case, you have like, I don't know, 2 to 5, this kind of sigma squared. And the first step is to estimate this like sigma squared, which is again variance component. And then once you know that, conditional on true parameters, so you can estimate fixed effect. And usually it's a way simpler case. The most of the time you spend on estimating variance component, which involves inverting some matrix, actually variance covariance matrix of your outcome you're modeling. The complexity is cubic on the sample size. And that's why like running linear mixed model is a way you spend a way more time than like linear models.
How did you convince lme4 to do less work? If it didn't have an option to specify this covariance matrix, it could only estimate it. How did you manage to use that package and supply your own covariance matrix?
I guess it's a really first equation of, or maybe the only equation in the article, but this kind of standard trick, which is I guess called decorrelation in general. But the thing is that you can, if you have a custom covariance matrix, you can apply Cholesky decomposition, or in a different case like eigenvalue decomposition, and represent as a product of matrix and its transpose matrix, and that really helps to stick to definition of random effect that is used inside lme4. Yeah, actually it was a kind of trick, and originally it was suggested by actually authors of lme4, and I know that in many publications some people just use this kind of custom code. So they just did once for their project, didn't share with anybody, and like implemented this kind of necessary step of including custom covariance matrix. And I was hoping like first year when somebody would do that, or like official release of lme4 will finally like implement this, because it was in to-do list, but it it didn't happen that time. And like slowly we start using our like custom code to do that within our group, and later on we created a package again, like to make it more serious software package with option to like testing, adding more stuff, sharing with other people. Yeah, and finally it ended up as a public package that now anybody can use it.
So I get how you use this decorrelation to transform the covariance matrix. So whereas before you knew it was some matrix, right, and you apply this trick, and now you know that the covariance matrix of the random effect is, I guess, the identity matrix. But how do you convince lme4, which is designed to estimate that matrix, not to do that and to skip that part and just to assume that the random effects are uncorrelated? Are you calling like the low-level functions yourself, and in this way you're avoiding the step where it estimates the covariance matrix?
Yeah, I would say it's a reformulation of covariance matrix. And yeah, you're right that what I did, instead of calling the main function of lme4, I just do it in 4 steps. They suggested to do that. And in some particular place, I just made this kind of reformulation, and useful for like if you have a given custom covariance matrix.
So what if you have these correlations induced by genotypes, but also you have these 5 different medical centers, right? How would you combine those two together? Because if you have genotypes, then you need to specify the whole covariance matrix and skip the step where it estimates the covariance. But if you have the medical centers, then you would want it to estimate the correlations between the medical centers. But how can you do those both things together?
Well, you just include two random effects, right? Like the model is itself saying that like random effects are independent, so you can just sum up variance, right? So if you just variance of two terms, it's a sum of its two variances. So, and basically you assume that one term due to medical center and another term due to like genetic relationship are independent. So, and you can just put two components, two random effects. So it's like called multi-component model. But still, if you suspect there is some correlation, I guess you can do that. In notation of lme4, it's called like random slope, or you can even introduce correlation between different components of covariance.
So even if they are not correlated, but I think you said that you estimate the whole covariance matrix and then just skip that step, right? So is it possible, like with your package with lme4qtl, is that possible to ask it also to estimate some component of the covariance matrix?
Yeah, sure. Like you can do it either in lme4 if you have like, let's say, 3 grouping effects and 3 random effects, or you can do it with lme4qtl where one of the effect can be like defined by custom covariance. Your variance covariance matrix of your, let's say, outcome is kind of, let's say, 3 or several components that you just sum up because they're like kind of independent. And this is the way you can model like variance covariance matrix of your outcome, which is multivariate normal distribution. You can easily do it kind of decomposition of your, let's say, structure and split it into like related to, let's say, one grouping which is medical center and another which is like genetic relationship.
And another feature of lme4qtl that you advertise is something called restrictions on model parameters. So could you explain what that is?
Well, in my case, we have exactly multi-component models and we have, let's say, 3 parameters and we were interested to test one of them. And the idea is the classical way in statistics to do that is just to fit 2 models, right? One is when you estimate 3 parameters and you have, let's say, in simplest case, 3 degrees of freedoms. And the other case, if you restrict parameter for your interest to, let's say, null value, which is, say, 0. So like there is no variance due to this kind of component. And so you restricting one of the 3 parameters to, let's say, value 0, and the other 2 are like free, you estimate, fit again the second model. And the way to derive a statistical test is just to compare 2 models, take their likelihood, perform likelihood ratio test, and you get a p-value. So that's just short story why I needed to implement these restrictions on model parameters.
And which parameters do you restrict?
So in my case, the model was modeling like a quantitative phenotype for like my data set, and in that data set, clinical data, I had like males and females, and you might think that there is some difference in, let's say, genetic architecture of this trait between 2 genders. So, and you might have one, like say, variance sigma squared contribution for males and another for females, and you might also have some correlation coefficient. So in this case, you have 3 parameters to estimate, and you just include them. But once you include it and fit that model, this kind of model is complex one, and you need to have a good sense whether it's worth to do that, and maybe just a simple model with one single parameter is a better way to model your data. So exactly what you do is just to compare your models, you just do kind of restriction model parameters, take this test, and you can quantitatively say whether complex model is better, like sufficiently better to replace a simple model.
So in your case, was including extra parameter worth it?
No, in my case, another reflection on this kind of dominance and additive modeling of genetic variance. In my case, doing a more complex assumption of genetic structure, there is some difference between males and females. It's worth trying, but usually people end up with negative results. So in my case, it was the same answer, that I couldn't get enough statistical significance to claim that there is some difference.
And I also wanted to ask you about the study that you talk about in the paper. It's called GAIT2, genetic analysis of idiopathic thrombophilia. So what was the goal of that study?
So that study was a clinical study. Clinical data were collected in Spanish families, 35 Spanish families. It was around 900 individuals. And these families were recruited with a proband of a person who had idiopathic thrombophilia. And this disease is called venous thrombosis. And something happened with your blood clotting process. I guess the prevalence is relatively low, which is below 1%, but still there is people suffering from it. And it's a disease which was previously shown has 80% of heritability, meaning that there is a good proportion of genetic contribution. And the project aimed to collect that data in families and try to find new genetic loci like QTLs that affect the risk of this disease. Project was done in St. Paul Hospital in Barcelona. The group that had a wide experience, I would say 15 or 20 years, MDs, biochemists, many people in the laboratory, that they were able to measure something like 500 phenotypes for this particular study, a vast volume of data, like diverse data, typical data analysis problem of how to make the best use of it.
When you measure multiple phenotypes, how do you combine them? How do you fit this model? Do you fit one model per phenotype, or is there any advantage to somehow grouping them and analyzing them together?
Yeah, it's a very good question, because the traditional way is as you said, is to model every single phenotype separately. And I guess what we did, but people try to combine and do what is called multi-trait station studies. And there is also an active development in this field. And even in one of my current projects, try to develop a robust method to do this kind of analysis. Yeah, usually it reflects how two given phenotypes are genetically correlated. When you say genetically correlated, it means that they can share some risk loci or QTLs that are common between given pairs. So you can include more than two phenotypes and try to do that. And actually, when you do multi-trait analysis, your typical null hypothesis is a given SNP has no effect for all phenotypes, which is kind of a different, more relaxed one. And once you find a loci, a QTL, which is affect your multi-trait setting, has some significant p-value, so you cannot say which one of the phenotypes is affected. So you can say maybe it's affected 2 or 3, or some portion. So it's kind of, I would say, an additional analysis, kind of complement to single trait GWAS, but still you can get insights on genetics of, let's say, a particular disease, but measured through different phenotypes which have a special name, which is called intermediate phenotype. And this way, how people try to enrich their data in just clinical setting, because a binary outcome 0 or 1 affected or control. So maybe it's a simplification, and it's kind of the final stage of the disease. But here you can consider many phenotypes and even modeling them, so you can just do a better job.
One detail of that study I found interesting, that you analyzed whole families, and families had, I think, at least 3 generations.
Yeah.
Why was that important? Why was it insufficient to have some cases and some controls just drawn randomly from the population?
Yeah, so there is this kind of family-based clinical study design. So it was widely used, let's say, 10 years ago. It was original study design. And the idea that you can find a particular variant which is cause the diseases, and it's enriched within a given family. So, and collecting data in families, you have a higher chance to pick this particular variant. And the problem is that the current problem of this family study design, that you are very limited, because it's very hard to recruit. So if you just see with the sample size of this GATE2 study, it's only below than 1,000 individuals. And on the other side, if you think about what you said, population-based approach, you can collect more data, let's say hundreds of thousands of individuals, or even approaching to millions of individuals. So, and for sure, the sample size is order of magnitude difference. And so your statistical power to detect a variant, it heavily depends on the sample size. And so most discoveries were done in this large scale studies, which is population-based. Still, family-based design are kind of useful, and you can see different kinds of specific effect, which is something like enriched rare variants or parent of origin effects. But I would say they're kind of secondary effect. Mainly, people just do on the population-based level, take data from as much individuals as they can, and just they find this strong effect due to large sample size.
So you mentioned some GWASs with large sample sizes, up to 1 million. If you were to analyze this, do you think lme4 or lme4qtl would do that? So you would have like a 1 million by 1 million covariance matrix?
Yeah, actually, it's a good question. And people who come to use my package in the GitHub, not a half, but a good portion of questions about the program goes very slow. And actually, the answer is that my package is based on sparse matrix method that implicitly assume some sparseness on your covariance matrix. And this is like due to implementation of lme4 that they basically based on, let's say, sparse random effects. In genetic studies, if you have families, I guess in the GATE2 study, you have only, if you consider this 1000 by 1000 covariance matrix, only 2% of entries are like filled by some numbers. 98% are zeros. Still, whenever the sample size is pretty like, if you run sparse matrix linear algebra methods, it's very efficient. And on the other side, like most data available, they're like population based, and like when some users try to plug in covariance matrices, which is thousands of individuals, so it's very computationally expensive because you just use lme4qtl package which originally thought for sparse matrices, and that's essentially not true for kind of dense matrices. I would say like there are many other tools that not only my package, but people can use it. But that's a clear limitation of lme4qtl.
Yeah, and even in your study, you sort of cheated, right? In order to get the sparse matrix, because you have all the genotypes, you could still correlate genotypes across families and get at least some correlation, right? It would be non-zero. But you specifically didn't do that to induce a sparse matrix?
Yeah, the thing is that once you have families, long time ago, people already know their like kinship coefficients. Imagine if you have a nuclear family with 2 parents and children. If you consider a single parent and a child, they share 50% of their genes, their genomes. So like the similarity is 0.5. And again, among children, they also have 0.5. And if you go further, and like let's say grandparents and like any kind of family relationship, so you can put theoretically computed number, which is like this kinship coefficient. And that's why it was not cheating. It was just to make use of this information that this family, and you just plug in the true kinship coefficient. Again, you can use genetic data just to compute this, and I guess we did it, and you just see that you mostly replicate this kind of non-zero entries, and where you have like 0 entries in like theoretical covariance matrix, you have some noise due to just finite sample size if you estimate using genotype data.
Sure, but maybe some of the recruited families were also related, like 2 families enrolled in the study, and they are in fact related because they have a common grandfather who already died.
Yeah.
Right, and you don't know about that relationship, but it is there. So in your case, your covariance matrix, where you assume those are zeros, those would be non-zero. So the admittedly smaller numbers.
Yeah, it's a good point, but I suspect it's like a negligible effect, but it might exist. You're right.
Is there anything else you would like to talk about?
Well, I feel that like our broadcast session was pretty technical, and something like mixed model might be complex if you never try to implement themselves. So at least my message would be that the package is ready to use, and these lme4 people, and generally like people who developed R statistical software, they make it easy to run like your own models. And I should say that based on like feedback from people who use my package, so they come from different fields. So, and if you feel you need some mixed model modeling, you have not only my tool, but any other tools available, even in R. So I hope if you play with your own data, you will not get bored with this technical explanation that we talked about.
Is there a data set that if someone doesn't have their own data, is there a public data set one could play with?
the time I was developing my package, I really wanted that dataset to appear. I couldn't find it. For example, you can just simulate data for yourself, but if you're really interested in real data, I guess there is some clinical studies that you can apply for like access to this. I guess this Farhingham study, which is happened here in Massachusetts. So you can just contact with people who like responsible for that study and get access to data. But it's not publicly available. You need to write an application form. And if somebody of your listeners like aware of this kind of package, so I will be happy to put a link on a web page and just maybe did some simple analysis with it.
Yeah, for sure. Cool, Andrey. Thank you for talking to me today.
Yeah, thank you so much for this opportunity. It was really a pleasure to talk to you.